So I'm going to give a talk that is really similar to a lot of the other talks that you've heard today, but it's dissimilar because I'm a different person. And uh, there you go. I've been working in strawberry since 2015. I did my PhD in strawberry. I graduated, I should say, went to seed industry for a year and a half, came back to strawberry as a postdoc, and I'm now an assistant professor and the director-elect of the UC Davis Strawberry Breeding Program. So in 2024, later in 2024, I'll be taking over the director title from Steve Knapp, who is my PI and colleague, and now, I don't know. Over the last 8 years, we've been really privileged to assemble a really high quality team of senior scientists and young scientists, staff personnel, graduate students, undergraduates, and everything that I've accomplished, that we've accomplished, is really attributable to them. So Steve is here, Glenn Cole is here. Glenn was the director of sunflower breeding at Pioneer Hybrid for 30 years. So when he joined the team, we really had two senior level professional scientists leading this program, which was really incredible. And so now it's, Wild, me, and we've hired Glenn's transition. This is Allison Krill Brown, who's formerly a small grains breeder in spinach and small grains, joined the team January 1st. We have a lot of supporters, collaborators, funding agencies. Some of those supporters and collaborators are also competitors, which makes strawberry just a really fun space to be in because you never know who's telling you what they actually know and who's trying to get your secret sauce. Of course, I want to thank the organizers of this. I was really surprised to get the invitation from David and Jeff. I really appreciate the opportunity to talk to everybody. Strawberry, like blueberry, like potato, like sweet potato, is a fairly complex set of conditions and logistical challenges that you have to balance out through the breeding program, times of year, and all this and that. I think we're lucky compared to blueberry, compared to tree crops, because our cycle is short. But we still have that generation where we're producing seedlings in a greenhouse, then we're evaluating material, seed propagated hybrids in the open field first look. And then from there, we're going to high elevation nurseries and propagating much larger numbers of plants for replicated trials across the state. So our numbers tend to be very tight as we move out of the seed propagated hybrids. So my mental philosophy is get as much out of the seed propagated hybrids as we can. And so one of the things I'll show you is some of the work that some of the stuff that I've been doing for the last 8 years effectively, and why I think that putting a lot of our energy in the seed propagated hybrids, phenotyping, genotyping, for us can be a good move. There's risk there, but I think all the things that we do are all going to come down to balancing risk and reward. One of those risks for us is the cost per acre in strawberry in coastal California. So in our target population of environments, we're spending $90,000 per acre to run a trial. That includes, fumigation, that includes the plastic, the drip, the human labor, and all of the data collection. Our trials, I think a lot of our trials in small fruits and in vegetable crops don't just happen once. We're harvesting in a day neutral trial, we're harvesting from March until December. So we might have 87 harvests in a year from that experiment. Our short day trials might run from December till July. So every month of the year, we're harvesting from multiple acres in our breeding program, and also trying to manage when and where are our plants going to high elevations, when are they coming to us from those elevation nurseries so that we can plant them for the next round of trials. The cycle is essentially 18 months. We are doing crosses in the winter. We are scarifying that seed and germinating in the following summer. We're planting out in the fall, and then we're looking at those hybrids for the first time in those single plant plots in the following spring. So our spring trials with our seedlings run typically from February to June. And then it starts getting too hot. We have to shift from fruit production to run production because we are a functioning nursery. And if you do some of the math in this, or some of the visual math in this short day phenotyping, day neutral phenotyping, and semi-plant phenotyping, as I said, we are phenotyping on-farm trials every month of the year and multiple times a week. This is a Gantt chart that Allison is starting to put together. And so as soon as you get out of that seedling stage, it just blows up. And this doesn't capture some of our more advanced trials where we have much smaller numbers and much larger plots, really those pre-commercial trials. So this is what we call phase 1 and 2. Things might stay in phase 2 for several years. Phase 1 is a replicated clone trial without the chill from the high elevation nurseries. If you linearize it, it's very simple. We make crosses, we evaluate things we don't really evaluate, but we have seedlings in the seed house or in the greenhouse, we plant them in the field. And then it's really, what do we do with that? Is that just a nursery that we're gonna throw things into the discard, or are we getting information out of it? So we're doing marker-assisted selection, whereas the mouse, we're doing marker-assisted selection in the 20K seedlings in the shade house to make selection decisions on what goes into that field. So those are things that are splitting our market segments with flowering time markers, eliminating things that are predicted to be susceptible to fusarium resistant or Phytophthora, and now we're introducing Macrofamina, or incorporating Macrofamina into that. We've been working on some metabolomic and metabolite markers that are published, that we're trying to basically validate the work from other labs in our germplasm. Some work, some absolutely don't work. Different teams have different quality genomes, and different quality genomes might have the same nomenclature associated with different homologous chromosomes. So you're calling homologs the same thing. And so there's a real mess in strawberry genomics right now that we're trying to nip in the bud. And then here as well, you're not trying to do high throughput phenotyping on on-farm trials, we're trying to do it here. You can imagine doing a yield trial on 10,000 single plant plots is very expensive. We can't do it. We can't get BRICS data on 10,000 single plant plots. And so part of the way that I'm thinking about this is, can we phenotype some portion of this using CD mean or some type of population optimization strategy here? So I'm not thinking about what data is that I currently have reflects this population, but what individuals inside of this population best reflect that population. So again, numbers, 20,000, 10,000. We pretty much go to 100 selections per market segment, and we serve 3 market segments. So as soon as we got it out of there, our selection intensity is already so high that if those are bad decisions for whatever reason, we're screwed. And then it's basically from there to product release is advancement. But that information is also coming back into which individuals are we going to put back into our crossing designs. But really what I want to do is come out of that 10,000 seedlings and be making crosses as quickly as I can, so I don't have a 4 or 5 year cycle, I have a 2-year cycle. Our barrier to entry, again, to building and to obtaining and accumulating historical data is our inability to do large populations and large sets of material within the target population of environments. I can really only do 100 things per year. That's not enough to train a really good model, especially if I assume that those things all have large fruit size. And so if I only have large fruit size things in my training population, everything's going to be predicted to be large fruit. And I might be able to segment out a couple things, but my predictive ability on those things that are decidedly worse than that are essentially going to be random guesses. And so prediction accuracy falls apart quite a bit when you have only your elite stuff in your training population. So genomic and phenomic prediction to increase population size, and can't really go beyond that 10,000, but it's how do we get better information on those 10,000, decreasing the cycle length, so making selection decisions right there as well as advancement decisions. This comes into that. Not every product has to be a parent, and not every parent has to be a product. So if you have a high degree of dominance, your best performers are not going to be your best parents. And so that's something that we're trying to capitalize and learn early. And then this managing the genetic variation piece. The way that I think about that is not so much in managing inbreeding across everything, but I have this target population for improvement. I have introgression crosses and breeding and sort of pre-breeding, pre-bridging populations. How do I make selections in these 2 different things with 2 very different goals at the same time using the same data but not just focus on GEBV. So I keep the best things and throw away all my introgression crosses that have individually no value, except for the genetics that they might contribute to the next generation. So they're never going to be products. This is an analysis that we did. We constructed a pedigree. It had about 9,500 things in it at the time, goes back to the 1800s. Across all of these generations and things that are connected, the average time between your two parents and you existing in the pedigree in strawberry was 15 years. So the actual cycle length on average in strawberry has been 15 years for the last 80 years. So that's way longer, obviously, than the 2 years that I said earlier, or the 4 or 5 years. But that's basically your first recorded offspring is 15 years on average after you were born, which is 70% of the average generation time of a human, which is pretty sad in a lot of ways, especially for a crop that is being propagated annually and goes into a sexual reproduction very rapidly once it comes out of a nursery. So we want to use GS or phenomic prediction to evaluate things earlier, cut out these really expensive trials, and cross sooner. So we want it to look like this, right? Every year we are making selection decisions. So right away I'm crossing hybrid 3 to hybrid 4 in my second year, and I'm pushing hybrid 3 into my multi-trial or my multi-environment trials. I have the usefulness criterion here. One of the things that we've tested, which is the sum of the genetic mean of a predicted cross and its predicted genetic variance multiplied by cell intensity. What we've learned through simulation and through the data that we've produced is these genetic variance predictions are so minuscule relative to the mean. So if you have variation in your means, the genetic variance in this estimator basically means nothing. So usefulness isn't really that useful. Whatever you can do to predict the genetic mean of the next generation is going to get you there 10 times out of 10. So even really high selection intensity going top to bottom, right, left to right, we basically don't improve or lose this really high correlation between the mean prediction and usefulness. And that might be a problem with the data. That might be a problem with the algorithms. So GS and this idea, it's great on paper, but what about in practice? We have been absolutely bottlenecked and hamstrung by genotyping costs. Costs have to be low, accuracy has to be competitive, turnaround time must be rapid. If we don't meet our deadlines, it doesn't contribute to decisions. It was a cost, it was just cost. And the data must be accessible. So we have to be able to use it in pretty much the raw format that's given to us. One of the things that I find kind of funny about the way that I think about genomic prediction and the way I think the animal crews are thinking about genomic prediction, I don't want it to necessarily predict my winners, my top winners, my best of the best of the best. I want it to help me to throw away the bottom 75%. I want to open up the bottom drawer and just like get it out. So if something, right, what seedling in my population of 10,000 things is going to be worth a $120 genetic evaluation, when 99% of the time it's going to be thrown away right then and there, right? I'm advancing basically 100 things per market segment out of those 10,000 things. So like a lot of people in the room, we developed a dark tag assay, which is on the right. And we also had developed this was SeqSNP or SNP-Seq HT, whatever the LGC thing was. They sunset that, and they didn't tell us that they were going to sunset it. And then they purchased Rapid. So we've now done a tech transfer to Rapid's FlexSeq. This is some of the results from DartTag. It works. The kinship matrices are essentially the same. The genomic predictions are essentially the same between the array, between the two DartTag platforms, and what have you. So yeah, Rapid and Agriplex have 500 marker platforms, which is a targeted GBS thing. These are $8 to $9 per sample, or less than 2 cents per data point. If you ever go to work in seed industry and you're doing single marker assays, they often don't think about samples, they think about data points. So that's why I have that there. They also have 5,000 marker offerings that are in the $12 to $16 range. Dart has a 3,000 and a 5,000 panel platform that are between, I think they're $13 to $15. They're less than $1 apart for the two of them. The arrays are exorbitantly expensive and obviously have the ascertainment bias issues and all of this stuff that comes along with them. But there's a 50,000 SNP array and an 850,000 SNP array. I actually don't know how expensive the 850K array is because we've never used it. We designed it and developed it with Affymetrix, but I have no idea what the cost is. And this year we've collaborated with GenCove to test out skin sequencing plus imputation. That basically you design or you do a bunch of WGS sequencing, high depth, you give that to them, they build out an imputation model, and then you recover all of the variants that are embedded inside of that WGS data. So for $36 a sample, we're getting 65 million variants with a concordance between 93 and 99%. So on the leave one out analysis, more than 90% of the genotype calls are identical between the skin sequencing plus imputation and the WGS data that gave rise to it. So, of course, we did genomic prediction. Sorry for the size of this text. We did GenCove. This is filtered by a minor allele frequency to biallelic markers only, blah, blah, blah. GenCove with SNPs, GenCove with variants just in genes. There are 2.3 million variants in genes in this platform, whereas our arrays have like 5 variants in genes. They're almost all intergenic, so it's hard to do some of the cool evolutionary stuff that you might be able to do. And then GenCove with just their indels. And then the FANA-SNP array, which is the 50K array, the FlexSeq and AgriSeq, which is 5,000 SNPs, and then those FlexSeq and PlexSeq, which Steve refers to this table as the alphabet soup of things. Going across the columns, we have yield, fruit count, fruit weight, fruit size, titratable or total soluble solids, titratable acidity, the sugar-acid ratio, anthocyanins concentration, skin strength, firmness, elasticity, and work, which are both integrals of skin strength or firmness. So how much total force is applied to get there. And in the top part of this, we have these models being applied to trained on seedling data to predict multi-year cloned blues, basically. And on the bottom, we were training on those blues and predicting similar blues. So it's just cross-validation. And there's really not a big change. So I want to talk a little bit about the genetic gain equation, not in a here's what it is. It's so right, but we've seen a couple of really nice examples of increasing heritability from very low values to very high values using image-based phenotyping instead of visual scores. And so you might really be able to increase your rate of genetic gain by moving to that. But if you already have a trait that's moderately heritable, you're going to be spending a lot of time and a lot of effort to increase the heritability. A 52% increase in genetic gain is all you're going to get if you increase your population size tenfold. So your effort is 10 times higher, and your rate of gain is 52% higher. This is a really nice paper. Everyone should study it. Gain is only going to be achieved when the target population for improvement, when the genetic variance is good. What does that mean? Well, it means if I have this elite population of things and I'm comparing it to a bunch of real crap, that's a bunch of genetic variance. I'm not going to achieve gain relative to my elite performers. I'm going to achieve gain relative to the average of the crap and the good stuff. So this isn't what we want. We want good genetic variance, not bad genetic variance. And so going faster actually is the easiest and most predictable and most direct way to get better. Going faster is just going better. So we took all of these approaches and we tried to estimate the rates of gain. This top number that I have here, this delta G sub 0, is the gain per year if I were to make selections based on seedling phenotypes only, no genetic information across this population of traits. The next one is I've now trained a genomic prediction model using 500 markers and those seedling data. The next one is I've trained a genomic prediction model using 50,000 SNPs in that seedling data. And then that last one is I've trained a genomic prediction model using 50,000 SNPs and the really expensive clone mean data. With all the seedling data, I'm going 2 to 3 times faster per year, which means in that same cycle length of my clone evaluations, I'm going 7.5 to 9 times faster. So my rate of gain in that same cycle length is excessively better, even though my genomic prediction and prediction accuracies are substantially lower. This is what I was so focused on in grad school, was like, how accurate are my models? And I would get excited about like, oh, I've gone from 65 to 75% accuracy. It doesn't matter. The only thing that matters is L. L is lifting this whole thing up. So that was, I think, really disrupting and upsetting to me, because I realized that the way that I thought about the world was wrong. And so then as I start reading these really excellent papers, the whole way that I've set up this experiment can't even allow me to capitalize on the best ideas. So this is sort of the classic breeding pipeline where you create a cross, you have, this is all with selfing and inbreeding going on, but you're doing these inexpensive phenotypes which is head GS, and that's my seedlings, or you're doing this conventional or this PYT, I can't remember what the P stands for, but it's a yield trial. It's some kind of replicated yield trial, or where you have fewer things and then fewer. So the population size, but the accuracy of the measurement increases very rapidly as you do this, because you're spending so much money. So what we've done is show that head GS is much better in terms of the rate of gain than conventional GS, which is exactly what this paper shows. And so I swear it's not results recycling, but that's what we showed. The single best method that has been explored in a couple ways by the same team is this 2-part strategy. And it's essentially where, and somebody talked about this in slightly different terms here, where you never actually phenotype your selection candidates. All of the data that you have is from the things that have been selected and have moved forward in previous years. So it's all using all of that historical data. We're finally at a place where we actually have a decent amount of historical data. We have 9 years of those field trials that I've been talking about per market segment. So we actually have a large amount of data, $300,000 worth of field trials a year. You'd hope that you'd have some data. So the next thing for us is to actually hit these 3 different ideas more directly against each other, and specifically the head GS, focusing on the seedling phenotypes versus, can I just use this really high quality, really expensive replicated clone trial data that I have to do in my pre-commercial trials? Can I use that to make genomic predictions? And is there a portion of that that I can allocate to updating my training model, right? If I'm only putting in the best, my training data is never going to have or understand what bad looks like. And it kind of needs to know that there is something worse than the best. Our target population of environments is expanding. It's still pretty small relative to maize, soy, and wheat. We're really focused on these gray dots here, but we are trying to move into the Central Valley of California, where there are more than 1,000 small farm stakeholders growing mostly conventionally, and they're growing varieties from the 1970s. So it's really essential, I think, that we do that. But those models are going to require different data because the environments are so different between coastal California and Central Valley California, the temperatures, everything like this. So we really need to know early which set of material are going to go to my short day markets, which are down south, and my day neutral markets, which are more up north, and then my Central Valley environments. And it might be that those are similar, that those are at least correlated. Everything's crossed. But we just don't know yet. So we need to be able to develop that data and then partition the populations much earlier, so that we're not trying to give these growers that grow on 1 to 3 acres maximum a 1.5 acre field trial to understand what variety might work for them on their farm. This is essentially my last slide, or one of my last slides. Again, this is that comparison of conventional GS in red, head GS in blue, and then the 2-part breeding strategy in the light blue and the pink from that paper. That's what I want to try, and we haven't tried, and we failed to build that into this set of experiments. So this is me dreaming in delta G. The next couple of stages, what we want to try to do is not necessarily predict means and variances, but look at multiple traits. How can I improve fruit quality and mean at the same time for yield and firmness? Our two market entry traits are really the size of the fruit, total yield of the plant, and the firmness of those fruits. We're a long shelf life, long distance shipping market essentially. But Brix is important, acidity, sugar acid ratio, and all these things are important. So what we're doing now is we have these predictions of positive correlations for yield and Brix, yield and acid, yield and sugar acid ratio with moderate to high estimates of yield. So 500 grams per plant in the time frame that we evaluated is 25 kilograms per hectare, 750 grams per plant is around 46,000 kilograms per hectare. So in a longer experiment, you get higher yields. For us in the day neutral marketplace, 100 kilograms per hectare is what we're after. So if we can accomplish those numbers and improve the average correlation within our families between Brix and acidity, then we can move acidity a lot faster without depleting the population mean for yield, because they are negatively correlated. So productivity, fruit quality, shelf life, disease resistance, without these, they're not going to go in the marketplace. Primarily because if they're not disease resistant, we got a ton of diseases. We got a ton of diseases in California, so we won't have any productivity. If we don't have the shelf life, nobody's going to grow it. If you have a big, beautiful fruit and a ton of them, but it's too soft to withstand the mechanical stress of packing and shipping, no grower is going to grow it, period. Fruit quality, we want to be happy about the products that we see in the marketplace, the products that we taste in the marketplace. So I was in a Trader Joe's the other day in California, and they're picking, we planted in November, they're picking already. This was maybe last week. The fruit looked terrible. So bad. And like that has nothing to do with me, and you can't see any you can't attribute any of that to me because my brand isn't on that clamshell. But it's insulting to see really high potential products, high quality potential products be treated in a way that is doesn't allow them to achieve that potential on farm. So yeah, this talk was supposed to be serious. I don't think I'm a serious person, but these are some of the really nice strawberries that we've produced. When you pick them right, they're beautiful. They taste great. They're resistant to disease. They withhold or hold up under pressure of packing and shipping. So there's a lot you can do with strawberry. There's a lot of things that can be moved really rapidly and really aggressively into what historically were closed breeding programs and smart decision making with introgression crosses and this type of thing. But yeah, strawberry. So thanks everybody for the opportunity. Thanks to all the supporters, collaborators, and funding agencies, the team again, and I'm happy to take a question or two if I have time. I think I have a lot of time.
You have plenty of time. Anyone dare question them?
Yeah. I'm glad I went through because I part of the challenge with working in strawberry at UC Davis is we're kind of in our bubble. And so we get these ideas and then they prattle around in our own little brains. So if anybody wants to challenge me, I want to be challenged. Jeff.
Yes, I challenge. Which publication is that from? Is that already out?
No. This is, I went too fast. This is a paper that I'll be submitting in the next week or so. Oh, same publication. Yeah. So we did this publication for this publication. We did this publication for the Dart tag platform because we're like, oh, it's gonna be great. I don't want to ship tissue to Australia because we've had couriers lose it. And so I'm really interested in working with a company that's based in the United States because there's less likelihood of them losing the samples in shipment. And we had started working on the SNP-seq or seq-SNP platform before Dart tag. And so as that sort of started going downhill, we started getting in bed with Dart. And then as these other things sort of started materializing, I was just like, all right, I don't want to be, the decider of which company has all of the access. So I just started sending out the target set of markers to anyone who would take it from me. And so we invested in Dart in terms of the development of this. We paid for the synthesis and all of the things for them, a third-party, vendor, to then go and make money. We spent probably $100,000 with LGC on their seq-SNP platform, in the development, synthesis, and testing of this thing, just for them to say, oh, we're sunsetting it. And so there's no opportunity for us to recoup that, right? They're going to be like, oh, here's your investment back, right? And we're not investors. So for me, it's like, if anybody wants to get in strawberry genotyping, I am happy to provide the genomic resources to get them there as quickly as possible and competitively as possible, because that's the only way we're actually going to bring prices down. Dart has no incentive to bring their prices down until somebody else can do it as well as or better than and is cheaper. So that's where I'm trying to get with Strawberry is everybody has their Strawberry genotyping service and they're all essentially equivalent. They all essentially have the same markers. So it's like, am I doing Illumina or am I doing ion torrent sequencing? It's like, I don't care. Like, yeah. So those two tables are in the same publication. And we're just burying all the genotyping platform development inside of it. So have fun looking for it. Yeah, Felipe.
Just regarding cost in the genotyping, when we compare the predictionability, the same table that
Yeah.
When you're using like 500 markers and 11 million of markers, predictionability is the same.
Yeah.
I'm assuming that you're going to use the left approach.
Oh, yeah.
In your breeding program, right?
Absolutely. And maybe with the imputation to something that's slightly higher density, right? At that point, we're paying for a 500 thing, but if we can capitalize on, the accuracy improvement that you do get from the imputation, maybe that's a better way, and you can eke out a little bit better from the accuracy. Sorry.
But I'm assuming that you're going to use imputation more for discovery than
Yes.
for prediction, right?
Yes. So for me, the GenCo platform has already replaced the array. The array is $58 per sample. It provides 50,000 data points, which is good, high density, reasonably high density, but I can't update it. I know that I miss genotyping individuals that have alternate alleles or second alternate alleles. And so they just look like homozygotes, but there's no way. When you go and look at your ability to predict a known parent from the genetic data and you have both parents genotyped, you would say, based on the metrics, that there's no way that's the parent, even though you know for sure that it is, because the entire purpose of the experiment was, is this the parent? So the arrays have, I think, a lot of issues. The GenCo platform, like any imputation platform, can be updated with new WGS data. So as we have these sort of test cross parents or testers that we're using in our breeding program as recurrent mothers, we're high depth WGS genotyping them and embedding them into this imputation algorithm so that all of those progeny are going to have higher and hopefully higher imputation accuracies. We were having this conversation earlier. I don't know when we start asking them to remove things from the imputation rules or from the imputation training model. I don't know if our imputation rule set would work for Driscoll's, would work for Vance at University of Florida, would work for other strawberry breeders. It works really well for us. It was designed to work for us. And so this entire idea is everyone can have their own cake and eat it too, potentially.
Normal timeframe, so the legal results.
The same time frame as DART. It's the same time frame as everyone quotes 4 to 6 weeks. We've got data back from GenCove, and that's everything that we do is tissue. We don't do any of the DNA extraction. So with them, they act as a middleman for a sequencing company, and then do the imputation and send you the data. And we got data back in about 4.5 weeks, which is shorter than some of the returns on CAST that we've had with our CAST service providers. So they're equivalent as far as I'm concerned right now. Yeah.
Grew up the game at all paper in 2017, but the part yes, yeah. I would sort of agree with you that this is me also one part of the other written example, but the one that studied. I think though it's compelling the simulation concept when the prediction model actually thinks with all the traits of the principle.
Yeah.
And I think that is my biggest reservation in practice, if you never will.
Yeah.
I think all the about. Instead of go through a two-part scheme, you only have the model guiding the blind. To me, that is something that I can't envision personally implementing in practice.
I think that's completely fair. For us, we have to plant out seedlings because we have to propagate them. Same thing with you. And so for us, it's those big traits. But if I go look at my seedling nursery, I am identifying things that have defects. Hey, maybe high yield, but the fruit are a weird shape, not the right color, this kind of thing. Those are discards. Still going to have to do that for all those other things. We catch those things 2 years down the line still. So I don't necessarily think that for a lot of those traits that appear under very specific environmental conditions, which for us seems to be the defects, and we don't want them to have that even potential in them. So we'll cut them as soon as we see one, even if it's been a high performer over multiple years and we've not seen it. So I think you're right, but again, I think it comes down to this risk reward piece. There is a risk to it. There is a reward to it. If the risk outweighs the reward, don't do it, right? If so much of the material is unpredictably defected, yeah, this is a really bad way to go. But if the majority of material does not carry those defects, or those defects appear in effectively one-off or randomly, then I'm going to take that risk because I can still make gain, and I'm not going to be probably throwing away an entire population of selections. Probably going to be maybe 10. I'm going to throw them away anyways, right? Of that 100, maybe one of them is going to go. So anything that's going to help me throw away those other 90, 99, that's just an easier decision. So that's sort of how I think about it. But with this conversation, usefulness, anything that's this plus this, and the part of it is variance, right? There's risk embedded in that because it's telling you something that you don't know, right? It's GEBV and the PEV. If I look at individuals with really good GEBVs or any GEBV and a really large PEV, it's not saying that that individual is bad or that the only opportunity for it is to be worse than its predicted value. It might be way better, right? And so I don't want to invest my entire next generation of crosses into these high risk individuals, but maybe some of them, right? So I think that there is a risk reward optimization in usefulness. There's a risk reward optimization in looking at PEVs. There's a risk reward optimization in terms of looking at the merit versus the diversity of my germplasm. So maintaining variance, maintaining inbreeding. And I think that until we start thinking about really what is the risk of doing it this way versus the reward of doing it this way. I think that it's, who knows? And we can probably come up, I could sit here and talk for hours about that. and so, yeah, maybe that's tonight.
Okay, well, thank you.
Thanks,
very much.
everyone.
Appreciate it.